Good evening and welcome, everyone. I'll just let you know there are some seats over here on the other side if you would like to sit for the program. So welcome. I'm Susan L., Vice President and Executive Director of the Foundation for Barnes-Jewish Hospital. Thank you so much for joining us in person and online across the country for Frontier Night 2023. This is an inspiring night when we get the exclusive behind-the-scenes look at the science fueling new discoveries and hope in our fight against cancer. And it's all possible thanks to your generous giving at our annual Illumination Gala and featuring the Cancer Frontier Fund. Now, many of you know Bill and Amy Komen. In 2009, they had the foresight to establish the Cancer Frontier Fund through our foundation to speed up cancer research so new treatments could move more quickly from the lab to the patient. The Foundation for Barnes-Jewish Hospital is proud to support Siteman Cancer Center and the groundbreaking research and expert patient care that is happening right here in St. Louis. Now, one way we do that, of course, is through the Illumination Gala. Tonight is the prelude to the Illumination, but it's also a celebration, a night of education, a time to reflect on how we can all make a difference in our collective fight against cancer. I'm so proud to introduce our lead ambassador for Illumination this year, Penny Pennington, who is managing partner of Edward Jones. Penny is one of the most dynamic and inspiring leaders in our community, and we're grateful she is sharing her passion and her leadership with us to spark more support for cancer research. Most recently, Barron's named Penny to the list of the 100 most influential women in the United States finance. Through her own personal journey with cancer, Penny has forged a powerful, heartfelt commitment to Siteman and to all cancer patients. Penny and our enthusiastic ambassadors are creating exciting momentum to make Illumination's impact on cancer research greater than ever. So now, help me kick off the evening, if you will join me, and let's give a warm welcome to Penny Pennington, our lead ambassador.
Thank you very much. Good evening, everybody. It is wonderful to be together on this gorgeous spring day. Susan, thank you for such a nice introduction. What we really are is the ripple effect of hope. Hope that comes through caring physicians, incredible scientists, and donors who believe in innovative research that makes a difference in people's lives. I am part of that story. That hope, that ripple effect, is part of my life for over 20 years, having been diagnosed with breast cancer. About 20 years ago, as a very young woman, 3 months after my mother was diagnosed with breast cancer. We went on that journey together. And 20 years ago, in some ways, seems like a long time ago, and in other ways, I remember every step of that journey so vividly. And one of the most vivid steps of that journey, and a real homecoming in a way for me this evening to be on the stage with this group, is that one of my first doctors in St. Louis, having moved from Detroit about 3 years into my treatment, was Dr. Kathy Weilbacher. And so I am intimately invested, my life is invested in this journey of hope, the ripple effect of hope. When I was treated about 20 years ago, I was treated very well between Detroit and a National Institute of Health Cancer Center there, and then moving to St. Louis. And frankly, that Siteman took my case is one of the reasons that I did move to St. Louis. I was treated well. I was treated with a standard of care and a set of protocols that were just right 20 years ago. But the difference that hope makes and that innovative research makes is that today I have a friend and partner who is being treated here at Siteman. She travels here from Michigan. She's being treated in a very tailored way, a way that's just right for her based on the diagnosis of her path and her journey. That's the kind of tailoring that our innovative scientists and doctors and the funding for that innovation can generate. Recently, the Foundation's Cancer Frontier Fund invested in a very significant discovery, a discovery about metastatic breast cancer, which currently has no cure. And one of its typical routes is to work its way into the bones. And I can remember being terrified of whether that might be part of my journey. Now a two-pronged immunotherapy approach to breast cancer, specific to its progression into bones, may be just about ready for clinical trial. It's that kind of innovation. It's that kind of help with the journey to prevent and then to defeat cancer that your generosity helps to fund. So many of us have walked this path before, and we want far fewer of us to walk this path, or if we do walk this path, to walk it in a way that teaches us all the blessings of this path without the devastation that sometimes occurs with it. So I am so excited to hear these marvelous doctors, these physician scientists, these compassionate caregivers, and be ever more proud of Siteman Cancer Center and what we get to do together. So Dr. Lynch, John Lynch, the president of BJH, Barnes Jewish Hospital, is here with us tonight to carry on our program. John, please join us.
Thank you, Penny. And man, what a great night, huh? Beautiful evening. The sun's finally setting over there in the corner. Thought we were going to have to hand out sunglasses and sunscreen. Nice to see you all this evening. And Penny, thank you for leading the charge and leading as our lead ambassador. But a leader means you have a team, right? Absolutely. We have a team of ambassadors here. And let me just tell you a little about their role. Our ambassadors are vital to engaging more people and sharing the excitement about Illumination and Cancer Frontier Fund and all the great work that comes from it. And you're going to hear some great stories tonight. If you are if you're an ambassador, if you're on Penny's team, please stand up for a minute for a round of applause. Thank you. They've made an extra commitment to help us with that personalized journey as we discover, as we prevent, as we treat, and as we cure. If you're also a board member for the foundation, maybe you could just raise your hand. Thank you. I see all of you. Thank you so much for your commitment. I know Tim has got his own stories about what he's seen over the last 35 years or so, but as a physician over that time period, what we are doing now and what our investigators and our physicians are doing in terms of prevention and treatments and cures and hope is just amazing. In fact, it was unimaginable for me what we see today. Really, truly unimaginable. And yet, I think all of you here, all the ambassadors, Penny and others, have imagined a different world as we fight cancer. And so tonight, we have that special look behind the curtains at what's going and what is imaginable, what's possible, and what the future will bring. At this point, I'd like to ask my colleague, Dr. Tim Eberlein, to come up and introduce our panelists and get the program growing. Thank you so much for being here this evening. Thank you, Tim.
All righty. I just want to take a minute to tell you how important what all of you are doing is in this effort. I get an opportunity frequently to talk about the research that happens at Siteman Cancer Center. And by the way, we have about $175 million of research annually. And it bridges every type of cancer. And that research is done by people like we have right up here who are world's authorities. And it was something that we established as a paradigm back when Siteman was first being launched. And that was, we wanted to be experts in every kind of cancer. But if you think about that, that's really a heavy lift. And now, 20-some years later, we actually are world's experts in all those kinds of cancers. And so you're going to hear some examples this evening. The other thing that we did is we recognized that to be able to replenish and establish new grants, do more innovative research, the only way we could do that is if we had a venture capital fund at our disposal. And that was really Bill's vision. Not only just to take money, unrestricted money, and invest it, but invest it in areas that would accelerate the pace of discovery. And more importantly, accelerate the impact on patients. And as Penny said, really establish hope for our patients. And so in my career, boy, I used breast cancer as a paradigm. I can remember as a medical student, we treated breast cancer in one way. Patient had a mastectomy. Radiation therapy wasn't very well worked out. And we had one chemotherapy, nitrogen mustard, which didn't work as it turned out in breast cancer. Now, 40 years later, what's happened? We have personal vaccines. We have personalized treatments. We've realized that breast cancer is multiple different diseases based on the clinical markers, etc. And oh, and by the way, in the last 20-some years that Siteman has been in existence, there's been a 30% reduction in mortality due to cancer. But there's one thing that's really unique about Siteman, I think, and that is all of you, Penny included. Penny, we couldn't do what we do if we didn't have these carefully invested dollars in new ideas. And that's what keeps generating the new research. And what has happened? In the last number of years, we've invested 120 projects, and we get an 8.5-fold return on investment. Most of that money comes from the National Cancer Institute, some from other institutes in the NIH. That's all because of you. And I think that's actually pretty unique to St. Louis. So give yourselves a round of applause. Now, tonight, we have some really interesting new projects that have been funded by all of you in the past. You're all familiar with prostate cancer, and again, I'm going to pick on my own career. When I moved to St. Louis, we basically did one operation for prostate cancer. You had an open radical prostatectomy, and patients had incontinence and they had impotence and other side effects. Now tonight you're going to hear about some of the really Star Wars imaging technology, because we realize not everybody has to have radical therapy for prostate cancer. And so it's a pleasure to introduce a person who trained here and was on the faculty back when I was chair. Eric Kim, Associate Professor of Surgery, R01 funded investigator, and he's going to tag team with Joey Bolito. Joe is an Associate Professor of Radiology. He runs our clinical research imaging program. And so Eric and Joe, take it away.
Great. Thank you so much. Do I stand up? Do I sit? Do I stand? Doesn't matter. Well, thank you guys so much for having us. I'm honored to, be able to share what we work on. And as Dr. Eberlein said, you all are instrumental, critical to any of this happening. So I thank you deeply, and I know my patients do as well. So I don't know if you guys are able to see the screen, but as Dr. Eberlein alluded to, we need better tools for prostate cancer. So PSA is blood test that's used for screening for prostate cancer. It's much maligned because it's not perfect. So how can we do better? So prostate cancer, it's extremely common, most common cancer for men in the United States, 200,000 cases in 2020, and now 250,000 cases this year, estimated to be at the end of this year. And PSA, a blood test, it actually does a good job picking up prostate cancer. It just picks up a bunch of other stuff too. So it's like if you go deep sea fishing with a giant net, you'll catch the tuna, but you end up catching the other fish, some jellyfish, some things you don't really want. And you end up doing a lot of biopsies, putting men through procedures they don't need. And so in America, we do about 1 million biopsies per year to diagnose those 250,000 cases. So the majority of biopsies are really unnecessary. And so how do we do better at actually figuring out who may have the cancer before we put a patient through a procedure that's pretty uncomfortable? So one thing that's come around over the past decade is MRI. Joe can probably tell you guys more about this than I can, and I think the first image, at least on my screen, looks terrible, so you really can't see anything. But if you look at the middle screen, there's the red arrow sign, which for us, urologists, which we jokingly call ourselves dumb plumbers, the red arrow sign is really helpful. We're not as smart as those radiologists. But you see, if the prostate is that gland in the middle of the screen, that bright white area is telling you that that tissue is not behaving normally. It's not the texture of normal prostate tissue. It's firmer, something that could be cancerous. And now MRI has become fairly ubiquitous. I mean, it's become almost a standard follow-up test to an elevated PSA. The problem is that MRI is not perfect either. So we've done a lot of the research here suggesting that MRI has its limitations. There's areas of scar, areas of inflammation that can look the same as cancer does on MRI. So the accuracy of MRI is about 70%, which if it was my uncle or my father, is not enough to say you don't need the biopsy. So it doesn't really get you to a threshold where we can change the paradigm. So what do we need? We need a non-invasive tool, kind of like MRI, that's objective and very accurate. And the accuracy is really what's limiting the clinical MRIs at this point. So diffusion-based spectrum imaging is something that one of our collaborators, Joe's and my collaborator, Victor Song, had been using in brain MRI, brain imaging, to help diagnose different forms of multiple sclerosis. Because taking a biopsy of the brain isn't so easy. So is there a non-invasive way to know exactly what's going on in the brain? And we thought, okay, well, why don't we apply it to the prostate and just see what we find? So instead of those MRI images, you get these heat maps. And the heat maps differentiate different components, different biology that may be contributing to the abnormal spot that was on the MRI. So if you look at the MRI image, that area again with the red arrow sign, for us dumb plumbers, is abnormal. And you say, well, is it cancer that's making up that abnormal area, or is it other stuff? And so image D is actually the inflammation component, the inflammation that the patient has. Image E is cancer. And so now we know for this patient, well, that area that's abnormal, there's some inflammation there, but there's also cancer there. So this guy probably does need a biopsy. But maybe in a different patient, it's just inflammation that's causing that appearance. So then we had some very intelligent, much more intelligent than me, postdocs that said, AI is this thing, this machine learning, artificial intelligence. Maybe it can be more objective than humans can be. Maybe it can do a better job telling us what's actually going on. So we trained an AI program to look at those heat maps, and we told the program what the biopsy outcomes were, what was the pathology. And so if you look on the left there, that's the conventional MRI. This one doesn't have the red arrow sign, so we're out of luck. But the DBSI heat map does tell you, there's a hot spot that looks like there's cancer there. And the diffusion histology imaging, DHI, which is this AI program, takes all those images and it spits out to us and says, there's a tumor here in purple. And not only is there a tumor there, we can tell you that we think it's probably a high-grade tumor. It's a Gleason score is the grading system of prostate cancer, but it goes from 1 to 5, the grade group. And we can say, oh, this is probably a grade group 4 or 5. And so this patient definitely needs some sort of treatment. And in our preliminary work looking at men that were going to undergo prostatectomy, this DHI program had 97% accuracy. And then for men who are going to get a biopsy, we found that it had 82% accuracy, which we think it'll do better, with more training. So the machine learning, just like my 15-month-old, the more training it gets, the better it does. So that's the future of what we're doing. And here I'll turn it back. But again, thank you guys so much for the support. We were awarded a SIP, pre-R01 mechanism in 2020. And we got our R01 in 2022. I don't know what the exact numbers on the SIP are, but the R01 was for $2.7 million. So it's a pretty, I think, good return on investment for you all. And again, we thank you very much. So I'll turn it over to Joe.
Okay. Oh, hardly. Thank you, Dr. Eberlein. Thank you to the Siteman Foundation. Thank you, Barnes Jewish Hospital Foundation, for everything you've done for us. And I think you got a good view of what Dr. Kim has been talking about. I'm going to talk to you about the next step where we're going beyond DBSI. And so I'd like to start off about this idea that we've had, Dr. Kim and I, kind of evolving over time, about how we can start to bridge radiology and pathology, these two different clinical specialties, into one. And so to start off, I'd like to talk about a very cool set of images here. On the left, we're going to talk about the evolution of radiology. The year is 1971. That man over there on the left is Dr. Hounsfield, the person who invented the first CAT scanner. And that's the first CAT scanner you're seeing here. And the image on the bottom is the first image of a patient that we've got. This person had a brain tumor. It's kind of hard to see, but that's not as relevant right now. But on the right, I think everyone knows what that image is. It's an iconic image of Abbey Road. Can anyone make an association between these three images? There's a clear connection. So not many people know this, but EMI Records, the people who signed the Beatles, took that money they made from the Beatles and gave that to Dr. Hounsfield to build the first CAT scanner. So it's because of the Beatles that we actually have medical imaging the way it is today, right? So it's the Beatles that bind us together, right? So I think that's actually kind of interesting. So what we're going to do is we're going to advance 50 years later. It's now 2022. We've got the flagship of imaging scanners, the 7T MRI scanner. And now because of the scanner, we are now able to see into the smallest structures of the human brain and any other structure in the body, the smallest blood vessels, to not only understand how small or how detailed these structures are in the human body, but what they actually start to do. So I'm in awe when I see these images. So now we've got this really clear development or evolution of how radiology has progressed over 50 years. Now let's talk about pathology. So here, this is one of my microscopes I actually got from the Path Department. This is around 1970. And now on the bottom there, you've got a glass slide of a tissue section. So basically what happens here is we take out a prostate, for example, and we cut very, very, very tiny, thin sections, and we look at those thin sections under a glass slide in the microscope. Now we advance 50 years. The technology overall is pretty similar, right? We still use these glass slides. But one of the major driving technologies that has really advanced pathology is the onset of genomics, right? So we've all heard about this, sequencing DNA. So now at the McDonald Genome Institute, we've got all these wonderful DNA sequencers that can now take the tissue on that slide and convert it into DNA sequences, which now we can now translate into risk assessment for the patient. If this patient with prostate cancer, they're looking at the DNA, does this patient have a high risk of getting recurrent prostate cancer? Does this patient have an intermediate risk of getting prostate cancer? Does this patient actually have a low risk of getting recurrent prostate cancer? So as a radiologist, I'm thinking, well, wait a second, maybe we can actually start to bridge all these fields together, radiology, omics, and pathology. And this gets into the work that I'm doing with Dr. Kim here. So now we've got this clinical problem, because once Dr. Kim removes the prostate, and this is an image of a prostate you're seeing on the left here, as it's sitting on the table in pathology department, it looks different colors because we put ink on it to determine left versus right. But this prostate is about 8 centimeters long. And inside this is a cartoon of what this prostate looks like in the middle. There's a brown spot there, and that's a prostate tumor, which is about, let's say, a centimeter. Let's just kind of ballpark it. So now if we think about it, how many slides do we have to make to look through that entire tumor? If the tumor is 1 centimeter tall, and each slide is about 0.005 centimeters, in other words, 1/20th the width of the human hair, that means we need to cut 2,000 slides of this tumor to look at the entire tumor. I will tell you that in prostate cancer, Dr. Kim will tell you this as well, there are certain parts of this tumor that are bad actors and other parts of the tumor that are not so bad. So which one of these 2,000 slides are we going to cut to send that for genomic sequencing over McDonald and actually figure out what is the best risk assessment for this patient with prostate cancer? What slide is going to give us the most valuable prognostic data? So this is how we typically do it. We will cut through it maybe a couple times and look at the key parts of the tumor and make a diagnosis based on the cells. But what we need to do is we need to now look at that entire tumor, and I'm going to show you how we're going to do that. Number one, with a technology called MALDI mass spectrometry, what I call molecular pathology imaging. And number two, DBSI MRI that you heard about from Dr. Kim, which we call molecular MR imaging. And so this is how this MALDI histology imaging works, and this is now available through the McDonald Genome Institute at WashU. We have a glass slide. So now the pathologist has just looked at that glass slide, rendered a diagnosis, and now hands me the slide. I'm going to take that glass slide and I'm going to put it into this machine here. And what's going to happen is that this machine is going to now fire a laser at that tissue sitting on that slide. That laser, just like in Star Wars, is going to vaporize that tissue and turn it into molecules. Molecules that can now give us prognostic information on this patient with prostate cancer. And now not only can we turn this tissue and cells into molecules, but now we can now say, oh, wait a second, we've got this molecule here that we know is going to be associated with a poor risk assessment for this patient. Now we need to do something differently. So these slides are kind of washed out, but now here's an example of something we're able to do now with pathology and imaging. So now we have this glass slide in a patient with prostate cancer, and the image on the bottom is supposed to be a prostate cancer. It's kind of tough to see, but I'm just going to circle where the tumor is. None of us can see where the tumor is, even if we saw the colors. But now I'm going to take that slide and put it into our MALDI mass spectrometer. Now, I think you can very easily see where the tumor is, right? You've got that nice, beautiful kind of circumscribed, that oval area that's red and yellow. Not only is this, and this is the computer talking to us, not a person. The computer tells us, hey, guess what, guys? There's a tumor there, but it's not only saying that, it's saying, wait a second, there's a tumor there, but it's expressing a bad actor molecule that's going to now have very, very major implications for our patient with prostate cancer. But it goes beyond just the slide, right? Because now we're talking about MRI. Can we actually slice through the tumor without even having to use a knife? So this is where the DBSI molecular MRI comes in. Now we can put that prostate into the machine itself and slice through with the MRI, and guess what? Now we've got a DBSI MRI diagram there of the tumor. It highlights it very nicely. That matches all of the prognostic molecules we've talked about. So this is giving us the ability now to non-invasively slice through a prostate, to tell us where the bad actors are, and to give us information on the prognosis for that patient. So in summary, this is what the future of prostate cancer at Siteman is looking like. We've got now a man who's going in for a clinical MRI scan. We've got our DBSI molecular MRI that now gives us a non-invasive risk assessment. And finally, these are the three words I want to leave you with tonight. Can we actually start to develop something called a virtual molecular prostatectomy that does not need scalpels or needles, but now gives us the ability to slice through a patient's prostate and the cancer, determine where the bad actors are, give us a risk assessment that we're going to now use to develop a treatment earlier on for the patient that's going to save his life. So anyway, thank you very much. And just also to add, sorry about that, a little premature there, but Dr. Kim talked to you about a $2.7 million grant that we got as part of this, but now the data I just showed you has been now recommended. It's within funding range at the NIH for another $3 to $4 million. So all the information you're seeing tonight is a pretty good return on investment, if you ask me. So thank you very much.
Not quite so quickly. I need to introduce you. All right, so the next two speakers are going to talk about breast cancer, but they're going to talk about breast cancer in a little different format. Over the last number of years, the National Cancer Institute has begun to hold cancer centers like Siteman feet to the fire, because you have these Star Wars kinds of projects that we impact the way we treat cancer patients. But what do we do about the cancer patient that has a hard time getting to our facility? What about the patient who can't afford their cancer treatment? And so over the last 20 years, Siteman has had the program for the elimination of cancer disparities. And our next speaker, Dr. Faluso Adamawia, we call her BC. I suggest you do too, okay? But BC is a professor of medicine, and she has not only done a marvelous job taking care of patients at Siteman in the big house, as we say, on Kings Highway, but also at our satellite in Christian Hospital, where she has made a huge impact in the patients in North St. Louis County and North St. Louis City. And so Dr. Adamawia, she's going to tell you about a project that she had done looking at genetic testing, especially in minority patients. So,
thank you for that introduction. So, what we have been working on is trying to improve the care for Black women with breast cancer by improving the rates of genetic testing in those who have been recently diagnosed with breast cancer. So, as you may know, genetic testing is a medical test that identifies abnormal changes in genes. And for genetic testing, you need just a little bit of DNA from human tissue. And that could be from hair, it could be from skin, from blood, or even from a mouth swab. And so if an individual has genetic testing, it can do a couple of things. So it can tell you where you're from, your ancestry, and if you have a risk of developing certain medical conditions in the future. And the nice thing is that some of these medical conditions have interventions to actually reduce the risk of developing them. So genetic testing is important for certain diseases. So there's some genes that we found that if you have a mutation in these genes, it can lead to a higher risk of developing breast cancer, like the BRCA1 or 2 genes. And you might have heard of some families having lots of breast cancer in them. There are also some red flags that lead us to suspect that there might be a mutation in the family, because these genes are typically passed on in the family. So if we see multiple individuals in a family with the same cancer, or we find a woman developing breast cancer at a very early age, or we see male breast cancer in the family, then we suspect that there might be a mutation in this family. So a couple of years ago, we found that a significant number of Blacks who had breast cancer were not getting genetic testing, even though they met the guidelines for testing. So there's a national organization that we follow their guidelines called the NCCN, and they have strict criteria for who should get genetic testing. And what we found was that under 60% of Blacks who should have gotten this test were not getting the test. And genetic testing is very important because it actually has implications for how we treat patients. So for instance, if a woman has breast cancer, the surgeon might recommend that she have a bilateral mastectomy rather than just removing the cancerous lump in her breast. If we know that she has one of these mutations, we might recommend a targeted chemotherapy rather than the garden variety standard of care that we recommend to other patients. And then it also has implications for family members, because if a woman has a mutation, she has a 50-50 chance of passing it on to her offsprings. So if we know that there's a mutation in the family, we usually recommend what we call cascade testing for the rest of the family members. So if Blacks or other minorities are not getting genetic testing when they need to, then this has implications for their care and also for the care of their family members as well. So we tried to figure out, what are the reasons why blacks and other minorities are not getting genetic testing when they meet guidelines. And what we found was that a lot of the burden, the fault is actually with us as physicians. So several studies suggest that we physicians are not recommending the test and referring our black patients to the same degree as we're referring white patients. There was actually a study from one healthcare institution somewhere on the East Coast where they looked at all their patients who were treated by the same physicians in the same hospital. And for those who should have met the guidelines for testing, 93% of their white patients got testing versus about 75% of their African Americans. And so what we did was that we polled medical oncologists and surgeons who treat women with breast cancer around the country and asked them what they were doing in their practices. And so what we found was that only about 60% of them agreed that blacks had lower rates of genetic testing than whites, and we expected this number to be 100% because this is, well documented in the literature. And we also found that less than 2% of those physicians who responded to our survey said that they were more likely to recommend a white for testing rather than a black, and we expected this number to be much higher. So what we felt was that there probably was some bias on the side of physicians in, selecting who should have this testing. So what we're currently doing with the SIP funds that you all contributed to was to try to identify barriers and facilitators that impact the recommendations for physicians. So if we know what are the barriers, to recommending a woman for genetic testing, perhaps we can address those barriers. And then the current study that we have ongoing is a study where we have a breast navigator in the clinics with the surgeons. And what she does is she does a preliminary assessment on who should get tested and then passes that assessment on to the surgeon in an attempt to help the surgeon identify who should be tested. And we're doing this to see if we can close the gaps and improve genetic testing for blacks with breast cancer and consequently improve their care and the care of their family members. So thank you for your donations.
Next is an old friend who actually both of us were in Boston together. I was a few years older than Kathy, but Kathy Wildbacher is the Langenberg Distinguished Professor and medical oncologist who does research in breast cancer looking at the environment, not the environment like in general, but the environment near a tumor cell and the impact that the immune system has in having a cancer grow, for example, as Penny alluded to, in bone. But tonight she's going to talk about something, again, that she's very passionate about, and that is, can we develop programs to improve screening and have patients who are underserved, who have less access to medical care, can we get them the same kind of care that we've been talking about? So, Kathy.
Let's see. Can you hear me? Thank you so much, Dr. Eberlein, for the kind words and for basically being our visionary leader, champion, cheerleader. He gives us pep talks. Yes, I've had many a pep talk. And he's just been a wonderful friend. And Kim Eberlein, friend to many here, also a champion of the cause. Thank you, both. And also, how wonderful to see Penny. We were both young, and we still look young. We're still young. And it's just great to see you. I feel so honored. As Dr. Eberlein said, I study the microenvironment of tumors, but This is our great, wonderful environment for Siteman. As you know, and I think you've heard, these SIP funds, the Siteman Investment Program, the Cancer Frontier Funds, they really help and inspire young, and you know, I guess I've been a recipient. I'm not that young, but I was young at the time. But young scientists and physicians, and they help our patients. They help catalyze a lot of great stuff. And I just want to say, let's see, I have a special thank you of something that accelerated my own research. 5 years ago, the wonderful Pat and Mark Burkhart, they were the co-chairs of the Illumination 5 years ago, and they and their friends made a generous Pat Burkhart Breast Cancer Research Fund. And I have to say, I got to be a recipient of these funds, of some of these funds, and I want to show you just one little glimpse of how this could work. This is I teamed up with I did a project that I'd been wanting to do but didn't have the ability to do. This is an electron microscope. So we were talking about all this microscopy. This is a single resistant cancer cell in bone that we've been trying to figure out, why are these things resistant to treatment. That what we see, that that's a single cell in blue. That purple stuff around, we found out, is a protein shield that protects them from the treatment. The stuff in the middle of the cell are basically protein factories. We then did some deep RNA sequencing and molecular analysis, and we figured out what they're making, how to target them. We teamed up with some chemists to make these nanoparticles that you can put the drugs in. We treated them, and it worked. So we're now in the process of trying to get to clinic. But really, without this kind of stuff, we can't dream and come up with cool stuff. But this segues to, again, Dr. Eberlein, and our mission here is the Cancer Center. I'm one of the co-leaders of the Breast Cancer Research Program, and we've known for a while that we have some great disparities in our region. And, you know, we are a cancer center. We want all of our patients to thrive and benefit, and we want to prevent cancer. And one of the things that that we lose sleep over at night, I would say, is why are these disparities? How can we reach everyone? So I'm going to show you a few sobering slides and tell you why I think what we're going to do about it and why I'm pretty hopeful about it. So this is a slide looking at breast cancer mortality, or dying of breast cancer, in St. Louis County on the left and city in the right, and the differences between Black African American patients in red and Caucasian patients in blue. And so you could see that there are disparities here, that more likely to die of breast cancer if you're Black in this city and county. And we took a little deeper dive and we looked at age, and we found that especially young women, you are 4 times as likely, if you're a young Black woman, of dying of breast cancer than a white woman in St. Louis County. So this is a problem, and there are multiple factors and many things that we're studying about this. Some biological, some social determinants of care. But one thing that we do know is that if you can catch a cancer early, you have a much better shot at treating and eradicating than if it's later. And this is a data for all comers of breast cancer, that if you could catch it at stage 0 or 1, the precancers are 1, we have almost 100% chance of curing and stopping the breast cancer and surviving the breast cancer at 5 years. And these are usually are mammographically detected. They're often not palpable, they're small, and thankfully that's where in our country most breast cancers are diagnosed. And whereas if you wait to the more advanced stages, the 5-year survival is less. Now, we have amazing treatments, many types of breast cancer. This is very general, but this is an opportunity. So what is our region? What do we see? So this is a map of our kind of the catchment. And let's look over on the left side where the darker red, and let me see how that looks. The left side is showing when patients are diagnosed with breast cancer, they're diagnosed at that advanced stage, stage 3 or stage 4. I mean, that means the cancer is larger and spread. And basically, I don't know if you all can see, but in the middle, the darker red is North County, North City, which is predominantly African American. And then just below that, there's Clayton, Mid-City, South City. That is lighter red and yellow. And basically, it's twice as likely to have a late stage of diagnosis in North City, North County, than Clayton, South City, Mid-City. Now look over to the green map. That's the chance of being diagnosed at an early stage. And again, now here, darker green is better. And you could see that North City, North County, and East St. Louis have significantly lower rates of presenting with an early stage breast cancer. So this is telling us we have something that we can intervene on, we can do. So we established, with the help and urging of our fearless leader, Dr. Tim Eberlein, we established what we're calling the Breast Cancer Equity Group. And I realize a lot of you are business people. It's not that kind of equity. It's Decreased Disparities Group. And we're trying to decrease the disparities in mortality through early screening, through risk assessment, kind of what Dr. Bisi was saying. You know, if you know that you have a strong family history of breast cancer, we need you to get screened. And then access to outstanding care. And we have learned, it's such a great environment at Siteman. I know you all know this, but it's truly an honor to work there. But we have people who are working School of Public Health, community engagement, where, for example, Dr. Drake is working with the community partners out in North County. We've got researchers on prevention, and BC is one of the leaders of our research efforts. We have marketing, head of radiology, Dr. Lannis Hall with outreach and education. And Meg Thayer is our program manager. I guess I'm the leader now, but it's really, we're a group, we're a team. And some of the things that we're trying, and so that's the team. Let me just move to this other great thing. So in 2019, Siteman established a satellite in North County. It's called Northwest Healthcare. And it's about 6 miles away from Christian, Northeast, Northwest Healthcare. They're both in the north. And basically, it's amazing. It's an amazing location, world-class doctors, surgeons, medical oncologists, physicians, scientists. We have a great nurse navigator. The community partners are now working closely. And thanks to the vision, we've got a genetics high-risk clinic, which is pretty rare and wonderful. And then the head of radiology is very dedicated. So we have this great site. We have a whole team. We've figured out what some of the barriers are, what's going on with the screening, what can we do for research, and now we've got to implement it. So our hypothesis and our approach is that if we design an environment where we can do personalized delivery of screening results. So when people come in to get the mammogram, first of all, you got to get them in. That's a whole other effort. But you come to get your mammogram, part of the problems is people are not, let's say you find something, the delays to getting a biopsy or even not showing up for the next steps are high. So we have this idea that we're going to, during the mammogram, and this is what we're piloting, patients get the results, right then and there. We will do a risk assessment, listen to family history, and determine if they would need to go to the high-risk clinic. And we have a navigator that will actually help right then and there get all this done. And we did a pilot just last week, and we have another one coming in 3 weeks, and it was great. We actually found 2 patients. One had a family history of male breast cancer, another ovarian cancer. They need genetic testing. it was known in the chart. And anyway, they were very happy to find out. We got them navigated to the clinic. Anyway, this is an idea of trying to actually change our model of care to meet the needs of the community. We're also trying to mitigate fears and distrust and issues by getting the right kind of information, working with our partners, working with marketing. We're trying to navigate women through these cost barriers. That's a big deal for patients. And we're trying to get our newest therapies and world-class approaches. So now all of our trials are being able to be opened at North City, North County. We have, I think, of all cancer centers, some of the highest minority accrual rates. But patients, if you offer the trials, they go on them, whether you're white or black. Patients love their doctors at Siteman, I think, no matter what. And so we want to make sure we deliver wonderful care. And just a couple of examples as I close of what we are ready to do right now. And thank you for this type of funds that you all can offer. BC alluded to cascade screening projects. We're trying to figure out where do we start. And one way is if we have a patient being treated that already has breast cancer, we can look at the family members and encourage them to get mammograms or screening if needed. How can we do? We've got a project for that all ready to go. Electronic medical records. We can get this medical history into the chart and find a way to trigger, wait, you need a mammogram. You need to go to high-risk clinic. One of our professors has a trial all ready to go. It was top 2 in the country for funding, but the CDC could only fund one because I guess COVID took the other money. Ready to go. Navigation. We know how important it is to navigate our patients to get the care that they need. And BC's got a couple of trials. We've got some trials ready to go. So I guess in summary, I would just say we've got these disparities in our region. No one wants that. Everyone wants this to be better. And we have, I think we have a huge opportunity right now to turn this around and to have a huge impact on our region and our patients. And we're very proud to be supported by our hospital and our cancer center. So thank you very much for all that you do.
Thank you, Kathy, and thanks to all our panelists for wonderful comments and observations today. Tim, I'm going to ask you to come back up and maybe moderate a few questions. I was watching Henry. Henry's always got the first question, and he was waiting. So, Tim, let me have you come up and take a couple of questions and work with our panelists here, and then I'll finish up.
Henry. Actually, it's a little more complex than that. So what we're really doing is picking projects that we think are going to have the maximum impact. They're different paradigm. They're new innovations. They're combining technologies like you heard about from Joe. There's no way that we can fundraise enough money to fund those projects. But we get preliminary data, we get information, and then we get funds from the National Cancer Institute. And over the last 5-7 years that we've been investing in these projects, what has come out of these projects? It is like a venture capital fund in the sense that they have produced new treatment paradigms, more personalized treatments for lymphoma, for breast cancer, lung cancer, etc. And that has accelerated the pace of development. So it's a whole process that we've put into place. It's a very careful, I use the financial world as an analogy. Before we carefully invest your research dollars, we study very carefully which projects are likely to work because of information that we know and because of our experience. And that's how we've been very good about investing in each of these projects and having them carry forward. So does that make sense? Other questions? Yes.
So I'm wondering if anything that's been going on in Missouri these last several years legislatively has either impacted research funding or recruitment or anything that, that we should be aware of.
It's a good question. Without being political, as I'm always supposed to be apolitical, there have been challenges. So as many of you know, Siteman and many of our investigators at Siteman lobbied very heavily to pass a referendum that many of you voted for. The state legislature actually did a very concerted effort at undermining that effort. And many of the patients who are eligible for Medicaid are exactly the patients that Kathy and BC were talking about tonight. And so, yes, we've had to overcome that. But it's part of this very comprehensive program that we've put together over years, and it has made a difference. So let me give you some statistics. One, in the last 8 years, we've reduced late-stage presentation in black women, 43%. So it's made a difference. If you look at all comers, white and black, in North County, we've reduced the late stage, stage 4 metastatic disease. We're now below the national average. We used to be 2.5-fold higher. So the programs that Kathy alluded to and BC alluded to in terms of screening, navigation, outreach, education, programs with the churches, with the Breakfast Club, with the Breakfast Club Brothers, other kinds of programs in North County has made a difference. Other questions for any of our panelists?
One of our viewers online for
Online.
Dr. Kim, if you
Perfect.
don't mind.
Oh, geez.
Dr. Kim, how far away are we from the imaging technology you described becoming the standard of care, and how can we as attendees tonight support that?
That's a fantastic question. No, so we're part of the NIH and NCI funding right now is to get that DBSI imaging before biopsy, and we're still just trying to recruit men to, again, train that machine learning AI platform. And we think it'll be another 300 or 400 men that are really required for us to know if the AI and the machine learning program is going to work. But I think once we've proven the concept, I don't see, especially as a non-invasive tool, what the barriers would be. That's something I don't have enough gray hair to know quite yet.
Maybe you and Joe would comment, because not only diagnosing these tumors with a non-invasive technology, but then using MR-guided ablation, that would be the next step. And so, you know, in a way, I use breast cancer as a paradigm. You know, we do breast conserving treatment, we do much smaller surgeries, more targeted treatments, et cetera. We're going to be able to do the same thing in prostate cancer rather than do radical surgeries. So I don't know if either of you.
No, I think that's a great point. I think part of the issue with focal therapy now, you're treating just part of the prostate. You know, the gentleman comes in, you get the MRI, you say, well, I don't know if that spot is actually cancer or not. Let's take you for the biopsy. You do the biopsy, and then afterwards you find out, oh, okay, that spot was cancer and the rest of the prostate is actually non-cancerous. Okay, well, let's sign you up for the subsequent treatment. You know, so there's more instances that you have to come in, more things that you have to have done. In an ideal world, if we, you know, get this technology working and it's very accurate, you can almost do the imaging, take the appropriate patient immediately to focal treatment. And in that regard, you know, we're aside from these projects, we have a number of clinical trials where we're looking at different, you know, energy platforms to try to treat parts of the prostate. You know, one that's really exciting is irreversible electroporation, something that's I think the liver surgeons have been doing for a while. And again, maybe us dumb plumbers weren't listening, you know, to the talk in the doctor's lounge. But compared to cryoablation or thermal ablation, there's no fallout zone with the electroporation. And again, that's an important concept for prostate treatment because the nerves are close, the rectum's close, all the real estate in the pelvis is you know, very high value. So.
Other questions? Henry.
Last question. So, the approval process for funding, I mean, I'm sure it's relatively easy to approve a person who's been around the block, has gotten some previous NIH funding, but what do you do for the brilliant young physicians who have their own ideas and maybe a lot different than the typical idea? How do you
It's really a great question. We've set up really a study section within Siteman Cancer Center to evaluate people who want to apply for an R01, people who want to develop team science like Joe and Eric, people who want to develop an elite team of very disparate experts to solve really tough problems like our pancreatic spore and our leukemia spore, et cetera, et cetera. But what we do is every time they submit an application, we give them a very detailed review. And the chair of the committee actually will sit down and mentor the applicant. Say, well, here's what is cutting edge, and here's where you're not quite ready for prime time, et cetera. And it helps mentor particularly the young people. In our cancer center, over 2/3 of the leadership of Siteman Cancer Center started out as a junior assistant professor. And in fact, every one of the individuals here who are leaders in our cancer center today started out as a junior person. And so it's something that Siteman does very, very well. And it's because of the environment that we have at Barnes Jewish Hospital, Children's Hospital, and Washington University School of Medicine. It's a pretty unique environment. So thank you for the question.
Thank you all for your questions this evening. I believe we have one more, and then I think we'll wrap up for the reception.
One more, and then the hook comes out. Okay.
Thank you.
So all of you, by the time they get to you, you know what they have, or you have an idea of what they have. So there's some genetic testing, some blood testing that's occurring now that indicates maybe some early stage cancers. Do you all see a role of that in your process or the workflow or in the future of treating these cancers?
What do you think? You know, again, biomarkers, genomics, being able to segregate patients into smaller and smaller, more personalized buckets, I think, happens. We've given you examples in prostate and breast, but I'm looking at Dr. Govindan, who specializes in lung cancer, and it's a perfect example. 5 years ago, 7 years ago, we used to treat lung cancer with chemotherapy, and the patients actually didn't respond very well. And over the last number of years, using biomarkers and genes, we get a personalized recommendation of a cocktail of chemotherapy, immune therapies, and the patients do dramatically better. That's just happened in the last 4 or 5 years. So these kind of personalized treatments are a result of our advocacy of and study of genes and biomarkers. And you've heard examples from our panelists tonight. John, are you next?
How about another round of applause for our panelists? Thank you all this evening. For our moderator, Tim Eberlein, thank you. I hope you guys had a glimpse tonight at what is going on across the entire medical center, across Siteman, across BJC Healthcare, BJH, related to the fantastic research that's being done by investigators like the team we have tonight. My observation tonight is that there are very few places in the country that can match, not rival, not beat. I picked that word carefully because there's really only a handful of centers like Siteman that can do the type of integrated research that you saw tonight, and that is really pushing back the frontier of cancer research, treatment, and diagnostics, and is now being recognized for that disparities work, which is so important for our community, and are being recognized around the country for the type of work that's being done to close those gaps and do the type of implementation science that needs. We frequently make amazing discoveries, but it takes 20 years sometimes to impact many, many patients, and then there are patients that are left behind that don't benefit from that impact. Our team's working on that as well. Very, very few teams can do the type of work that you see today. One last observation, that team needs a leader, and for the last 25 years, it's been Dr. Tim Eberlein, the executive director, and working with your colleagues at Washington University. So, Tim, thank you very much for leading the team. Behind the scenes, always encouraging. Penny, can I leave you with the last thoughts?
So being a former financial advisor and leading Edward Jones, this language of investment and finance makes a ton of sense. It resonates in terms of what we're trying to accomplish. I heard words like assets, equity, venture capital, early stage investment. I even heard about diversification, because this center is one of the most diversified cancer centers in the world, and that, Tim, this aspirational view, we want to impact every kind of cancer, all kinds of cancers, so that we can help more patients. Kathy, you talked about Siteman being such a great environment, incredible assets. There it is. People, knowledge, capabilities, teamwork across this integrated, a kind of integrated medicine that really makes a difference in more people's lives. So all of this innovation is funded by people who believe in sound investing, and that's us. And I heard all kinds of reasons to make that kind of investment. I heard about return on an investment in hard dollars. $1 turns into $8.50. I heard about that kind of sound investing, but I also heard about the kind of investing that changes lives and makes families and people whole, not to have to go through the devastation of cancer. So these physician scientists are so deep-hearted, they're so compassionate, but they do something I can't do. They look at cancer at a molecular level. I also know that they look at cancer at a human level, and that's what I can do. I can look at cancer at the human level, know what it's done to the life of my family, myself, my friends, my partners, my colleagues, my community, and know that I want something different for all of those people. So we can invest in defeating cancer at the human level and at the molecular level by making these tremendous investments. So I'm going to do what I can do and work with our ambassadors and all of you to say, give. So we heard this story tonight of the Beatles to EMI to the CAT scan. I mean, that's pretty remarkable. What's your story? What's your story about how you're going to align your investment decisions with the values of your family and with what you want to see different in the world? And I think what we are all aligned around is wanting to see a difference in kicking cancer to the curb. And this remarkable organization helps to do that. So what can we do? Well, we can give, develop that story, Beatles to EMI to CAT scan in our own lives, make decisions about how we're going to make investments and ask others to do the same. Please come to Illumination on June 3rd here at the Ritz. It's going to be beautiful. Enjoy the circle of hope that we'll find here tonight and at Illumination. Enjoy Colin Jost of SNL fame. He's going to be terrific. We're going to have a great time at Illumination and leading up to it. I want to thank all of you for being here tonight. Please go check out the foundation website for all kinds of other inspiration about what these incredible doctors and the Siteman Cancer Center are doing. If you are here in the room tonight, stick around. We've got lots more questions and wonderful people who can answer those questions for us. And if you've joined us online, thank you so much for your commitment. Thank you for being here tonight. Everybody have a great rest of your evening. Enjoy.
Hi guys, I can steal those from you. Thank you so much.